Hello, and welcome to this week's episode of Medisodes. Today, we're going to be talking about one of the most important reactions within the body, respiration. So there are two types of respiration, aerobic respiration, which requires oxygen, and anaerobic respiration, which doesn't require oxygen. So if we start with aerobic respiration, we have at its core an overall reaction of glucose plus oxygen going to carbon dioxide and water. It's quite simple, and that's probably what you learned at GCSE. But let's go further into the different stages of respiration. So let's start with glycolysis. So glycolysis, as the name suggests, is the breakdown of glucose from primarily glycogen stores into pyruvate. So there are two main steps. So firstly, glucose is phosphorylated to form two molecules of an intermediate sugar which has 3 carbons, and this is known as GALP. And this basically activates the glucose so that it can be oxidized. To do this, so 2 ATP molecules are used, and that creates 2 ADP molecules plus the 2 inorganic phosphates that become incorporated into this phosphorylated glucose. So you might be thinking, if we're trying to make ATP is our universal currency of energy in living things, why are we starting by using it up? Well, that's because in the second step, these 2 3-carbon intermediates will be oxidized to pyruvate that releases 4 ATP molecules. And that means glycolysis has a net yield of 2 ATP molecules. This process, the second stage, is known as substrate level phosphorylation. 2 protons are also removed from each of these intermediates, so that creates 4 protons overall, and these are used to reduce a coenzyme called NAD, and that forms 2 reduced NAD molecules, which are very important later in respiration. So if we look at the net yield of glycolysis, is that you've got 2 ATP molecules, you've got...
The next stage of aerobic respiration is called the link reaction. In this stage, pyruvate from glycolysis is converted into acetyl coenzyme A, and this takes place in the mitochondrial matrix. First, pyruvate diffuses from the site of glycolysis, the cytosol, across the mitochondrial membrane and into the matrix of the mitochondria. In the mitochondrial matrix, pyruvate is decarboxylated and dehydrogenated into a 2-carbon molecule, acetate. As this happens, a molecule of carbon dioxide is evolved, and NAD is reduced to NADH. This acetate molecule then combines with coenzyme A to produce acetyl coenzyme A. This molecule is then used in the Krebs cycle, the next stage of aerobic respiration. The Krebs cycle also takes place within the mitochondrial matrix. The acetyl coenzyme A from the link reaction acts as a carrier for the 2-carbon acetyl group. It reacts with a 4-carbon molecule oxaloacetate to form a 6-carbon molecule citrate. The coenzyme A molecule is now available to be recycled back to the link reaction and pick up more acetyl groups. The production of citrate allows for the Krebs cycle to begin. Citrate is converted into a 5-carbon molecule by decarboxylation and dehydrogenation. 1 molecule of CO2 is produced, and NAD is reduced. This 5-carbon molecule is again decarboxylated and dehydrogenated, producing a 4-carbon molecule. Here, 1 molecule of CO2 is removed, NAD is reduced, and ATP is produced from ADP and an inorganic phosphate group by substrate level phosphorylation. This 4-carbon molecule is then dehydrogenated again to produce another molecule of reduced NAD, and FAD is also reduced to FADH2. No decarboxylation takes place. These reactions regenerate oxaloacetate, which allows for the cycle to continue again. The net gain of the Krebs cycle is 2 carbon dioxide molecules, 3 reduced NAD molecules, 1 ATP molecule, and 1 reduced FAD molecule. For each molecule of glucose, the Krebs cycle occurs twice, because 2 molecules of pyruvate are produced from each glucose molecule. Hence, we have to double the yield for each glucose. Thus, from 1 molecule of glucose, we get 4 carbon dioxide molecules, 6 reduced NAD molecules, 2 ATP molecules, and 2 reduced FAD molecules. However, it may seem that the 2 molecules of ATP that we produce from this Krebs cycle is rather insignificant. All these stages simply to produce 2 molecules of ATP seems rather wasteful. And we also produce plenty of reduced electron carriers. Throughout the link reaction, Krebs cycle, and glycolysis, there are several decarboxylation dehydrogenation reactions, catalyzed by decarboxylase and dehydrogenase, and the reduced electron carriers that I mentioned earlier somehow need to be dealt with, and for that I'll hand over to Anupam.
Following on from the Krebs or citric acid cycle is oxidative phosphorylation. This occurs using the electron transport chain, which occurs on the inner membrane of the mitochondria. The inner membrane is folded into cristae to maximize the surface area available for oxidative phosphorylation. In the first step of oxidative phosphorylation, reduced NAD and reduced FAD, the 2 coenzymes, are oxidized back to their normal NAD and FAD forms. This releases hydrogen atoms, which then split into hydrogen protons and electrons. The electrons move down the electron transport chain. The electron transport chain is comprised of electron carriers within the inner mitochondrial membrane. These take the form of proteins. As the electrons pass along the electron transport chain, they lose energy. This energy is used by the electron carriers to pump the hydrogen ions also generated from the oxidation of NAD and FAD across the inner membrane into the intermembrane space. This creates a concentration gradient of hydrogen ions, where the concentration is higher in the inner membrane space than in the mitochondrial matrix. The hydrogen ions then diffuse back down this created concentration gradient back into the matrix via the ATP synthase enzyme on a stalk particle. This generates ATP from ADP by phosphorylation. The movement of H+ ions across the membrane generating ATP is also called chemiosmosis. The synthesis of ATP is important, as this is what is the primary purpose of respiration, to generate energy in the form of ATP. At the end of the electron transport chain, the electrons then join with oxygen from the blood and H+ to form water. It is often said that oxygen is the final electron receptor of the electron transport chain, as this production of water at the final stage is where all the electrons and hydrogen ions end up. The electron transport chain and the whole process of oxidative phosphorylation occurs multiple times across the entire inner mitochondrial membrane, and with hundreds of mitochondria within each cell, this process generates almost all of the energy the body requires. Out of the roughly 38 ATP that is theoretically produced from the whole process of respiration of one glucose molecule, the vast majority, 34 of those ATP molecules, will be generated via oxidative phosphorylation. Oxidative phosphorylation, however, does require the presence of oxygen in the cell, and which is why anaerobic respiration is often used when this is not possible. I'm now going to hand over to Shrey to wrap up today's episode and summarize respiration.
So, I mentioned at the beginning that there are 2 types of respiration, aerobic and anaerobic. So we've been through aerobic, and I hope you understand that now. So let's look at anaerobic quickly. So, anaerobic respiration actually starts the same way as aerobic respiration with glycolysis. So that means overall that a glucose molecule is turned into a pyruvate molecule, and also that two protons are released from each pyruvate molecule that's made. So these protons are then used to reduce NAD, that coenzyme we talked about earlier, to form reduced NAD. Pyruvate, the pyruvate molecule that we made, is then accepts electrons and protons from the reduced NADP, and that means it forms a lactate molecule. So, and then this process repeated of NAD being reduced to reduced NAD, and then giving away its protons and therefore reducing pyruvate to lactate. So, what's the net yield of all this? So, there are 2 ATP molecules. Those come from the glycolysis process itself, and 2 lactate molecules. And these lactate molecules will go on to form lactic acid. And what this does is, the lactic acid will lower the pH in your muscle cells. It's an acid. So after a while, as you do more and more anaerobic respiration, the pH will drop lower and lower, and eventually it will reach a point where the pH is out of range of where your enzymes can function. So basically, the pH will start to inhibit the cell's enzymes, including those involved in glycolysis. And that means eventually that anaerobic respiration will basically happen so much that it stops itself. So obviously, to keep going, we have to remove this lactate. And to remove the lactate, it's done in 2 ways. It's done by the liver. So the liver converts it back into glucose in a cycle called the Cori cycle. And also it's oxidized to pyruvate again if O2 then becomes present again. So that means that how it works is that the lactate is dehydrogenated, so those hydrogen ions are removed, and basically that pyruvate can then go into aerobic respiration as normal. But obviously for that you will need oxygen to be reintroduced. And how this actually works, anaerobic respiration works, is that it's usually used at the very start when you've just started to exercise, because it's at the point where your the concentration of oxygen in your blood is not very high, but your muscles are working need a lot of energy. And this creates an oxygen debt that you build up. As you exercise, you'll use more and more aerobic respiration, because that's a preferred way for your body to produce ATP, because it doesn't produce the lactic acid. And once you finish exercising, if you've been exercising really hard, you'll know that for a couple minutes after, you're still breathing really heavily. And that's so you can oxidize the lactate molecules that are built up in your body. And this is known as repaying the oxygen debt in your recovery. Both anaerobic and aerobic respiration are both used during exercise. And they're both really important in the body. So I hope you learned a lot more about aerobic and anaerobic respiration, about the glycolysis, the link reaction, the Krebs cycle, and oxidative phosphorylation, as well as anaerobic respiration processes. And I hope that that's given you a better understanding of how the body produces ATP, which is the universal currency of energy. Thank you for listening to this week's episode of Medisodes, and tune in next week for another episode.